about the server, how to access, but actually I think it's quite clearly written in the email. So all those people who asked for access to the server, I sent a detailed description how to connect. If you haven't got it yet, but you want to work on this server, just drop me an email and I will send you the details. And yeah, finally I decided not to make separate users for everyone. That's how normally it should work. So if you log into a server, you have your own user account, you have your own user space, you have your home directory, which is separated from everyone else. But the problem is that we have quite limited space in this server. It was even quite expensive just to have 200 gigabytes. And 200 gigabytes is not that much. So if you think about those data files we will work with, it's not huge. So that's why we have only one user for everyone, except me. I have a different user account. But we have one user account, and what we will do, we don't have to install all the programs for all the users. So we only need to install each software or application once. But we will work, or actually you will work in separate folders. So you will have your own workspace where you can make your own scripts, where you can make you can see your own results, or even where you can just work on anything you want, because the server is available every time. So you can log in outside of these courses as well. And if you have something you want to run, just feel free to log in and run on this server. We should use it as much as we can, because there is no no extra charge for using it, more or less. So that's why I actually decided to work this way. But that also means that we can see each other's files. So don't put anything which you feel is sensitive or not public. Of course, it's only us, so only the course participants who can see this. Only those people who got that SSH private key I sent out. That's the only way to log into the server. There is no other ways. That you cannot log in with username and password, which is a bit more common login version, but it's the private key is actually much more secure. I already checked the logs of the server, and there were hundreds or thousands of hacking tries already. So there are just these automatic robot machines on the internet that are looking for new IP addresses, and if they found something, they just try to hack automatically with different password and username combinations, but none of them will work. But even if anything happens with the server, if somehow someone logs in and do something nasty which shouldn't be done, or if we install something which will damage the file system or the operating system, there is absolutely no problem. The whole thing is virtual, which means that I can anytime reinstall the whole server remotely. So if something is happening within a few minutes, the whole system can be reset, and we can go back to our original configuration, which also means that we will lose the applications we installed, but they can be, of course, reinstalled. Okay, then I just log in and show you how the file system looks like, because that's, I think, one of the most important thing. So I'm now sharing the screen. I guess there is no problem by putting these up on the internet. I'm just thinking if there is any sensitive information I will write in, but I don't think so. And I will just simply share my desktop one. Okay, and now I can resize it. So you will see. Can you see it now?
Yes, yes.
Okay, so you can use, so it's on your machine, on your Mac or Linux machine, not on your Windows machine, but if you have your Mac or Linux, you can just add this private key to your private key pool by saying SSH add, and then go to your private key, which is, I think, here for me. Yeah, so like this, and say enter. I won't do it again because I did it already. But if you do this, you don't have to use that -I thing at the end. So then you can just simply log in by saying SSH bioinf at and the IP address. The reason why I'm logging with this one, because this is, so there are actually two different users. So if I'm logging with this user, I have the sudo rights. The sudo means that I have the super user rights for this account, but there is no, of course, there is no super user rights for the bioinf. So there is, of course, a different private key for this one. So we are on the server, and in the same way, if you have the Mac, you can use the CyberDuck. If you are going into something like this when you start your CyberDuck, there is this like watch icon here, which is the history, and usually you have your other servers here, so you don't really have to start again with open connection and blah blah blah. If you set up the connection once, you know, the SFTP, the IP address here, the username here, and the private key here, and then you click this add to keychain box, or leave it just ticked, then it will be saved, and then it will be in your history. So this is mine for the bioinf. I think I also have it for the Ubuntu user, maybe not. But I only have to go to this watch the history and double click on it. And it will, of course, ask me for the fingerprint. Okay, so I will allow the fingerprint. Oh, and there's a thank you txt already. Okay. So, but I don't want to show you here the file system. I want to show you the file system here. On a Mac, you just simply drag and drop files here later. But what we will see soon, we can also see something here like the user folders. But as I will show you now, so if you say df-h, which is always one of your very first commands when you log into a server. So when you, and this is not just in this server, but anytime you are logging into an in-house server which you are sharing with someone else, or even if you are logging into your virtual machine on your computer, and you start doing something, like you start building up a new pipeline, or you start analyzing a new data set, this is almost always the very first thing you should do. Check if you have enough space. Check if you have space for your raw data to copy on the server. Check if you have enough space for all the temporary files you will need. Of course, you have to know what you will do, but this is what we will go through during many examples here at the course. So in there are quite a lot, like almost 10 different like drives here. So each line represents something like a C or D drive on a Windows. So they are virtually or physically different parts of the file system. But you don't really have to know all of them. You don't really need anything on those on which are starting with TMPFS. These are all system requirements, and you don't really have to worry about them. There are two things only you have to check if you log into this server. The one is the usage of the main root. So this is the root directory where all of the folders are, and your home directory as well. So if you say pwd, you will see that you are in /home/bioinf. I'm in the Ubuntu, of course, because I'm in a different user account. But this is one of the things you have to check, especially if you want to, on this, in our server, if you want to install programs, which sometimes means that you are installing the databases as well, it will go to your, more probably to your home directory. As soon as you are starting using Conda, Mini Conda or Anaconda, Conda is always putting everything into your main home directory, the applications. So there has to be enough available space. So as you can see, there is almost 60 gigabytes we have in our home directories. And I think we will put, as I said, the applications here, but the data, for the data, we bought as a separate hard drive or a separate virtual hard drive, which is the other thing you have to check always. So when you log in, and you say df-h, you should check this if you have enough space to install your programs, and then you should check this, the Scratch directory, which is now completely empty, but there again, you should make your own user directory and check if there is enough space there. It's a bit bigger. This is the one with the 200 gigabytes of space. Again, this is not huge. So as soon as we will start working with genome raw data sequencing, raw data files, this can be filled up very easily. But I will try to provide you like smaller data sets every time we are doing working on something. So I think we can manage to keep enough free space here. Okay, so that's almost the first thing you have to do. The other thing you can do, especially if you are not working on the server when we are at the course. So let's say that you didn't have time to follow the live course, and you are just repeating the things based on the YouTube recordings, and you go up. You should always check if someone else is on the server first, and then if someone else is doing something on the server. Because if someone else is doing, then you won't see who. Because if I'm now say W, which is the command to see how many people are inside, as you can see, there are many bioinf users at the moment with the different IP addresses, of course, and these are all you who are now logged into the server, and also myself here with the Ubuntu user. So you will see yourself, and you will see if someone else is logged in. But anytime if you have questions, just please interrupt me. But it doesn't mean that even if they are on the server, maybe they don't work on the server, they are just there, they are just left their terminal open and they don't do anything. So the other thing you have to check, especially if you want to run something which requires resources, is the top command. So if you say top, you will see if there is anything running on the server. You should check usually the CPU and the memory requirements. So if anyone is here and running a program, it will require CPU time. This list is usually ordered by the CPU. I think in default it is ordered by the CPU time. So as you can see, nothing is happening. These are all system processes, and the highest CPU usage is 0.3%. So it's nothing. And also, if you push the button 1 here, then you can see that this one is collapsing or opening. So here you can see how many CPUs we have free. So it can happen that a user is logged in. You see that there is 100% CPU here running on a program, but that means that maybe just one. If it's 100%, that means that one CPU core is in use 100%. But it means that you have 3 other CPU cores you can work on. So if you see that there is some kind of activity on the server, but there are free resources, then feel free to do your job, feel free to practice. But if you see that all 4 cores are used, all of them are 100%, then just log out and come back a bit later, because probably if anyone is working on the server, they are just working for minutes or for a few hours maybe. Yeah, this is also something what I would like to ask from you, not running like days jobs here, because then we cannot use the server together. Okay, so this is the third thing you have to check. So always check for the free space. Always check for.
Wait, Lars, just show me the 100% again.
Here or in your computer.
On your computer, please. Thank you.
Oh, there is no. What you mean? How the 100%?
100%, yeah. Where do we check to see if it's 100%?
Oh, it's here in this
Okay,
the
percentage
CPU, the
okay.
CPU column. Yeah,
Okay.
the memory is also it can be used as well, but the most important thing usually is the CPU. The other thing which can go up easily is this WA, which is the I/O wait. So that's the input-output waiting time. So if someone is, for example, working with a huge data file, zipping, unzipping it, or just going through a huge file, copying something, it is usually using the storage quite a lot. So even if the CPU is down, but you see a huge number here, that means that it is probably slowing down the server significantly. So you should wait until this number is going down as well. If this is high, that's good, because this is the idle. So if this is high, that means that nothing is happening on the server. Okay, so the main things what you have to check is this column here. If this one is closed, like this is the default look for the top. But if you push the button 1, it opens. So here, this is the CPU usage. It's the same as this one, but separately for all the 4 different cores. And this column with the I/O wait. These are the most important columns you have to check. And if this is low and this is low and this is low, then you can just go forward and do your job. Pressing Q to quit from this one. You can also check the free, which is showing you the memory. I don't think there will be any cases when we will work with a program with a huge memory requirement. Maybe on the de novo assemblies we can fill up the memory. But again, this is something which can be used by jobs. So we have 24 gigabytes of RAM. It's not too much. So we have to plan our jobs or our subjects accordingly. Okay, so that's when you log in. Always check if someone else is here and working, and try not to interfere. If you want to install something, just because, I mean, as I said, we have only one user account. So as soon as I will ask one of you during this course to install Blast with Conda, for example, that means that if like Chioma installs the Blast on the server, it will be ready for all of you. So when we will start doing real bioinformatics and we will start with installing the programs, I will always ask one of you to do it. Or maybe I don't want to do, because I did it quite a lot. But I will ask you to do it, but only one of you during the course, and I will just, ask someone else next time. So that means that you usually don't have to install programs. So that's the next thing. If you come back and you want to practice, or if you come back because you watched the YouTube video, but you were not here, then probably those installations are already done. I will try to repeat this every time when we start installing programs, that before you install, just check if it's there. So now if I'm saying blast n, then it's not there, of course. Actually, the Linux is very kind and said that if I need blast, then I should install the blast by using the Linux built-in package manager. So this is when I think, I don't know if you remember, but one of my very first, in our very first meetings when we spoke about how to install programs, few of these very famous bioinformatics programs are actually the part of the Linux distributions. So this means that you can install the NCBI BLAST by using the APT, which is the Linux own package manager. So this is the same program you should use to update your Linux or to install the programming language environment. And some of the most famous programs are already included in these. Of course, if you want to install Snippy or a less famous program, you will probably use Conda. But just check this. So if you have to use blast, first check if the blast n command which you will use is present or not. If not, then install. If it's installed, then use it. Okay, that's very important again. We shouldn't try to install everything again and again.
Yeah, sorry, Laya. So I just tried that and it says, please ask your administrator. Do we always have to ask you each time we need to install?
No. So even if you look for Blast can be installed by Conda as well. I think there are a few programs that we will use much more frequently. I think they should be installed. So again, if you go back to that meeting when we spoke about this, if you are sharing a server within a department or within a research group, I think the best practice for those programs that are used by many people many times, like the Blast, they should be installed, I think, by the super user. But as soon as you are going down to your own project, to your own small job, I don't think it's good to do it for the whole computer, especially because it can also interfere sometimes. So there are a few bioinformatics packages that are that requires specific versions of another program, and if I install version X centrally on the computer, and you need version Y, then maybe your program will not work because version X is installed centrally. So I think in 99% of our cases, we don't want to install programs centrally. For this kind of command, if I want to run this, yes, I need super user rights. That's why the sudo is here. So the sudo means that what I'm doing, I will do as a super user. So let me just show you an example. If I want to check the root directory. So the root is the super user, and it has a directory, and I'm not allowed to do this because I'm not the super user, but my user has the super user, the sudo rights, so I can just repeat the same command and list that folder by saying sudo ls/root, which is actually empty, but this is done now. If you have a virtual machine and you are using your virtual machine on your computer, the the first user you set up in a Ubuntu file system is always the one which is having the sudo rights. So you probably have the same kind of sudo rights there. The bioinf user, what you are using, doesn't have the sudo rights, because you can also make a bit of mess with the sudo very easily on the server. So that was the third or fourth thing I think is very important. Also try to keep your job and your works in your own folder, and because you are all logging with the same user, it's very easy to share your work. It's very easy to say, okay, I made this script. If you want to use, if you want to look into, it's in this folder, this file name, just check it. And if you want, copy it, but never write into or never modify someone else's script or program. I think this is a quite normal way of behavior, but just because we have the same user, we have the chance to delete someone else's files. That's the other thing. Never keep any files on this server if you don't. I mean, you can keep your files here. You can analyze your files here. You can do anything you want, but you should always have the backup for your files, especially your raw data, because anything can happen with this server. There is a chance that we are installing something which messes up the operating system, as I said, and we have to reinstall. The good thing is, if we go back to the file system, if we need to reinstall the whole server, this one, the /scratch, will remain intact. So at least the files or results we are having in this one, and this is why I think it's very important to work in this directory, will remain there. So if we go to that, to the root of the file system, and I just say ls-l, so these are all the folders within the server. And as you can see here, we have, for example, the root folder, which can only be accessed by the root user. We have the home directory here, where you have your /home/ubuntu or /home/bioinf folder. And we have the scratch folder here. And I made the scratch folder accessible to everyone. So I don't know how much do you remember these things here. If this is RWX for everyone, that means that everyone can access this folder, everyone can make a subfolder, everyone can write, read, and execute anything inside. So if you go to the scratch directory, at the moment there is nothing, just this default Linux folder, which is created when you are mounting on a hard drive. But this is where you will make your own directory. It's up to you how you name it. If you want, you can name it with your username, I mean, with your CRS ID if you want, but you can also just use your name and say mkdir like this, and then you will have your own folder. You can also go there with your CyberDuck or FileZilla or WinSCP. Just here for us, you are going up to the root folder and go to the scratch folder, and I will see my folder here already. There is no other folders yet. You can always push refresh, and you just simply drag and drop your files here, drag and drop your files out if you want to visualize with your R. And this is the next thing what you have to do, always know. So you will work on data. You will have results. You may have a few scripts, R scripts, or any other programming language scripts, which you may edit locally on your workstation. So anytime you want to do something on the server with your script, you have to synchronize it. You have to put up your files. there are different programs and different solutions for this, and I will try to show you some later how I do this. But to be honest, even I wasn't able to find the ultimate solution. On Windows, I had a very good program, the Notepad++, which is a very good script editor program. So the difference between a text editor and a script editor, let me just see if I have one open here. I will have one open so I can show you, I hope. So if I, for example, if I go into my folder and use the Nano, which we used before, that's a terminal-based text editor, and I want to make a new script, which let's say it will be like per script at the moment. And I'm starting writing the script. Let me just, actually the Nano is doing some kind of syntax coloring. That's good. I will be very fast. Okay, that's what I wanted to see. So actually it is now colorful, which is good. So you can use Nano, and it will make the script colorful. This is what I wanted to show you with the script editor, which I hope I can bring it here to you. Can you see my? No, I don't think so. Okay, you can see it now. So this is how, for example, a real script editor looks like. So this is my all the per scripts on the HPC, for example, and this is one of them with a nice syntax highlighting. Also, script editors usually memorize your variable names, like when you do it, for example, in R. So if I start writing $path, as you can see, it's already here. So it's much easier to write the same variable name, much harder to mistype your variable names, which is very useful if you write a program. In many other script editors, you also have some helps on function, what are these and those functions do. I think we will go through a few of them. There are many free ones. This is called Sublime text. It's not bad for Mac. For Windows, I definitely recommend Notepad++. It's a very, very good script editor program.
Can I just say that with Sublime Text, it's actually free. The Sublime Text too, but you're gonna get the annoying message every
Yes, yeah. After like every 10th savings, you will get a message. I'm trying now, bringing up. I want, of course. Oh yeah, this is it. So this if you want to purchase or if you want to donate, you have this annoying message sometimes. I think I will collect a few of these editors for the next next meeting. Actually, I will collect them and I will send you the links in the email, so you can just look into it. This is again something which is, I think it's much more about your personal preference. So maybe you won't like the Sublime text, maybe you won't like this kind of color scheme. It's all about you. So you should try a few. I will give you a few tips in my next email about those programs I used. I also have one favorite on a Linux graphical user interface now. I don't remember the name, but I will check it later. Right, now I will put this here. Are you seeing my console now? So my simple desktop. I hope so. I think so. So anyway, you can also use Nano because it has some kind of syntax highlighting as well, but it won't give you the those extra functions. If you use Nano, you are doing your editing, Control O to save and Control X. So you are doing your editing on the server. So as soon as you modify your script, modify your pipeline, it is immediately there. If you are editing your files on your own machine, you have to use your file transfer program to update the script. Or there are a few of these script editors that can handle remote folders. As you saw in my Sublime, I had my HPC folder there, but it cannot really do it through the SFTP. Sadly, there are a few programs. I think Notepad++ can work through SFTP. So you just set up the same kind of login details, what you set up to your WinSCP or CyberDuck. But what I did, I paid for the Mountain Duck, which is brother of CyberDuck, and Mountain Duck is a program that can mount folders, remote folders. So if you look at my Finder, I have the HPC here as a folder here. So it looks like a local folder, and I'm using this as an almost local folder. But it's actually mounted through the Mountain Duck. That's why there is a duck there. Okay, so that's one of my tasks for you for the next meeting, to write a list on editors, and then you can just try them and decide which one you like. And of course, if you want to be a real geek or nerd like me, then you have to change your background and use the black background and the white text, not the white background on any of these editors. But usually you can't do it, even on RStudio. Okay, the next thing I would like to discuss with you, and I think this is enough about the server. The next thing is, do you have any questions regarding the server, how to access, how to work on it? If no, then I would like to go. I have a list, a very big list actually, on my ideas on bioinformatics project we can go through. Most of these are simple one meeting projects, and we can go through. And the way how I imagine this from next Tuesday is that I will make a few slides, like 5 or 10 slides in the beginning about the whole theory behind that particular bioinformatics problem, and then we are going through a real-world example and do exactly what we should do with our own data. So I have a few, quite a few ideas, and I would like to discuss all of these with you. So if you think that one of these should be discussed with a bit more detail, or you think it's not that important, or you would like to put something else inside a topic as an extra, then please just stop me and discuss. So I would like to ask you much more, having a discussion now than just me speaking. So because there are quite many people from my group here, I think we should start with some very simple assembly pipeline. So we should go through a de novo assembly pipeline. This will be a prokaryotic de novo assembly pipeline. So I have never done de novo assembly on a huge eukaryotic genome, and I don't think I would be very happy to do that or teach that. It's way much more complicated because of the size, because of the the different chromosomes and the diploid nature of the genome as well. So it's a bit easier with prokaryotic genomes, and this is something what you will have to do. I think most of those people who, especially those who are working in the vet school or around the big lab. But I think for those who are not working on this topic, there will be many interesting parts, I think, which should be learned. So there will be a Lenovo assembly. Is there anything as special you want to discuss or you want to hear about here?
I think it would be good to go in a little bit detail in the reference mapping, because
that's my next point.
Okay.
That's my next point, actually. And I think the reference-based assembly should be a bit more, maybe 2 different meetings, because there is a reference-based mapping, or we can also call it like reference-based assembly or reference-based realignment. So when you are doing a next generation sequencing and you have the reference sequence and you just want to compare your results to the reference sequence, there are different reasons why you should do this. And I think we can also speak about some eukaryotic thing here. So I did actually doc whole genome sequencings, and I'm very happy to show you how I did it, because that's almost like doing a human genome sequencing. It's a little bit different from what we are usually doing with a prokaryotic reference-based assembly. But also, if you are thinking about, for example, SNP, what you just simply using as SNP, but actually during the SNP, what you are doing is reference-based assembly or reference-based realignment. And there is also a good reason why you do that. So I think I would be happier to split the reference-based assembly to 2 meetings, one for the prokaryotic things and discuss all the aspects. Why should we use reference-based assembly? And also one for the eukaryotic. I think I will give you a kind of comparison between de novo and reference-based assembly somewhere in the very beginning. So that would be, I think, the start. Here, I'm thinking about doing metagenomics as well. Are you, Koto? Are you here?
Yeah, I am here. Yeah.
Because I'm thinking about doing your seal samples.
Well, that would be very cool if we can use that, and yeah.
So I'm sure
learning
that we have to do de novo metagenomics there, but we don't have any high C, so we have to do, I think, that kind of approach when we are using the genetic properties like coverage and GC content and K-mer contents. And because you have multiple samples, as far as I remember, how many samples you have?
So for the metagenomics we use, I think 10 or 15, I cannot remember. I haven't.
Wow, that's perfect. That's really good. Is there any difference between them, or all of them are the same sequence?
So 5 of them come from Antarctica and the rest is from Juan Fernandez.
But they are all the same sequencing depth?
Yes, they are all the same.
Okay. Yeah. But this is perfect. I think this is a very good project,
yeah,
actually.
I would be happy to do that.
Okay, we can do it together. That's a very nice, because this is this is an exact example when we probably have no reference sequences. So I mean, the extent how the seal fecal microbiota is researched, I think.
And I have for some of those samples, also the 16S version. So I don't know whether that could be interesting to also compare different.
Yeah, I think we can do some kind of
mixed
comparison. Yeah, I would like to do that as well.
Yeah.
Okay, that's good. I'm also thinking about digging out some human metagenomic samples, just to show you the huge difference between something we know and something what we don't know. So I'm pretty sure that for the human metagenomics, we can do reference-based methods because we know much more about the human microbiota. Okay, that's very good. Metagenomics then also ticked. The next thing what I really want to cover is sequence search, which means that that's something which is completely universal for DNA or proteins. So if you are coming from the protein world, it's the same very useful tool for you. So we will actually discuss 3 different approaches. One is the way how we should do how we are doing BLAST, but not just BLAST. It's like BLAST, BWA, bow tie. So that's kind of approach, which is nowadays quite the same, which means that you are looking for a small seed sequence and then you are extending that. The second thing is the hidden Markov model based sequence search. So that's what you should do when you are, for example, looking for a protein domain, and you are not just looking for sequence homology, but you also have weights on your amino acid positions. So I will speak about this, but of course there are conserved amino acids in protein structures, and if they are there, they represent a much higher match weight than any other residues. And the third one should be the pattern-based search. So when you are looking for an exact sequence pattern or something what, for example, we did with Ibrahim, I think I would be very happy to bring here your SNP search, Ibrahim, if you don't mind.
Yeah, of course. Yeah.
That's something what, again, you should do. If you know how to do, it's very easy and very simple, and it will give you the results very quickly. I'm also thinking about Jenny's. Should I call Jenny or Jenny?
Jenny is the official
Jenny,
way,
okay,
but
okay.
Jenny is the English.
Okay, Jenny.
Kind of.
Jenny, okay.
Yeah.
Jenny is microRNA data, which is again something what you will maybe meet with. I'm thinking about discussing that microRNA together with the TRADIS pipeline because it's a little bit similar. So in TRADIS, what you do is also, if you are going into the details of the TRADIS pipeline, you have a consensus sequence, which is a primer or a transposon sequence, and then you are looking the sequence which is after that known sequence piece. So this is something what you would do with your pattern search or exact sequence search, and then you are processing the data. Also, there is a project which I did together with Gingy. Gingy is not here today.
I'm here.
Oh.
Lajos.
Ah, you are here. Okay,
Yes.
perfect. So okay, so Gingy is here as well. That's so good. All of my listed works people are here. That's so good. So I think I would be very happy to discuss your project as well. But I think I would discuss as a separate full meeting project if you don't mind. If you are not feeling that it's confidential. I don't know, how do you think about that? So if because there are so many pieces in that project which is like very nice bioinformatics, I would say.
Yeah.
Okay.
Yeah. Great.
So it's again something which is a lot of sequence-based, pattern-based search, but also there are so many post-processing what we can do on that data. So that's an interesting thing, and it's actually a virus genome project. Okay, that would be the next one. So the sequence search. Maybe this will be more than just one meeting. I am thinking about doing some protein meeting as well. So I would like to give you at least one protein structural biology workflow. So if you need to figure out something on your gene of interest or protein, and then what is the is that workflow I usually do. So it's again a bit more subjective from my own experience, but because I did this for 8 years before I came to Cambridge, I think it's a usable and useful workflow, especially if you find a gene with unknown function. Then maybe the way to know a bit more about it is to go into the structure or the function. I'm thinking about maybe discussing this in one meeting. Just think, if you have a protein sequence, if you have a gene that you don't know anything about, what are the ways to figure out how to know a bit more about that. I hope you don't mind doing some protein work as well. The next thing is electrophysiology, and I'm not sure that we want to do this, but if you are interested, I have a lot of electrophysiology data. This is electroencephalographic data. We can learn a few very basic techniques on it, and also we can learn big data handling on it. I wouldn't say that this is a top priority, but this could be an interesting point. Is there anyone here at the moment who is doing electrophysiology or behavioral studies? So anything which is like connected to animal something? Not at the moment. Okay. I will put this as a low priority. Then I would like to go through the TRADIS, because half of our group is doing TRADIS. I think it's a very interesting technique. It's a very interesting pipeline. And again, there are some key points where you want to transform your data, a few points where you want to know more about a gene. So again, I think, if you don't mind, Ibrahim, I would go with your data set, because we know that there are some unknown genes at the end of your pipeline or your data, and then we can follow up with try to find out.
Yeah, that would be a great idea, actually. So we never really did it together in the end. So.
Yeah. Right. The next thing I would like to do is Arijit's CRISPR project. He's not here at the moment, but he's also following the course. And he asked me to do this quite a long time ago. I didn't have the time yet, but this is a very interesting thing. And also, I think, something which can come out in many of your future works. So that's a CRISPR-Cas system where he's inducing mutations in cell lines, and then of course sequence all these monoclonal cell lines. And what he wants to know is what's the difference between the cell line and the original wild type cell. So actually, this is again knowing the primer sequences and knowing the sequence environment where the mutation should be, and then find out how to automatically extract mutation information. It's a bit like the TRADIS, but it's a bit more, because it's eukaryotic. It is sequencing from both ways. And also, we don't just want to find out what was knocked out. So the TRADIS pipeline, in the TRADIS pipeline, we are knocking out genes, but we don't really care about how. But in a CRISPR-Cas system, you can introduce SNPs, you can introduce in-frame insertions and deletions, which means that you don't completely knock out the gene or knock out the function, but maybe you are introducing an interesting change in your sequence, which is interesting to follow up. So that's the next thing. Gingy's project is here. Oh yeah, there is one very important thing, I think, for most of us. Even I have to learn a bit about this before I'm teaching to you, is to make distance trees. So how you can measure distances between genomes, how you can measure distances between genes, how you can cluster them later on. So it will be the basic methodological meeting when we are going through all the different types of clustering methods, how you can use them in R, what are the inputs, and also how can you generate the distance data you will use. I think this is a very important thing for all of us. I am thinking about doing some kind of protein structure simulation as well, just to show you that it's not rocket science. And maybe, again, you will need this later on. This will be a bit more theory and a bit less practice, just simply because the practice cannot be done in minutes. Usually protein structural simulations are running for days or sometimes weeks. But I think it's very interesting to speak about it, and also very interesting to see how in like very small simulation, how it works. We had a very nice work together with my part 2 student last year with Lana, which is a complete workflow starting with bacterial isolate sequences up to the point where we actually defined the MLSTs, the locus sequence, the locus sequence types. So that's again a lot of assembly, a lot of comparisons of different genomes. I mean, a lot means that a lot of different ways. So how you compare full genomes, how you extract full SNP difference data, how you extract core genomes. So again, not just assembly, but then gene annotation, RORI, PROCA, going through all these steps. How can you visualize your core genome? How can you visualize your accessory genome? I think these are also very useful skills for all of us. This can be, again, more than just one simple meeting. And as I said, I would be very happy to go through Jenny's microRNA workflow as well. So these are my ideas on the paper. But if you have any other, I'm very happy to hear that. Or if you would like to learn something which I haven't mentioned, just please tell me. Also, if you have your data, what is sitting on your computer, and you feel that you need some help to analyze it, we can just do it together. If you don't, if it's not a problem that you have to share your data, of course. For many of these meetings, I will be able to provide the data. But if you would like to discuss your own project, come up with that. Even if you don't say it now, just send an email to me that you have this or that kind of data and you would like to go through, and you are happy to share. It would be very useful.
Lajos, so I have been working with my 16S data that I analyze. Well, I clean the sequences with KIME and stuff like that. But I have come to like a growing sense of lack of confidence, because I don't really have the criticism or like the critical background to actually know or look at the files. Because you should, like people that are not like that knowledgeable about informatics, you just follow a pipe. And it would be very cool if we could like do some sort of like criticisms on like outputs and how we can recognize when an output is like looking not correct. Because usually the pipeline are built with like data that always work, but the problem is how can you identify when something is looking not right, and then yeah, when can we find the cues to actually go again and then see what's not, like, yeah, how we can improve that?
Yeah, this is a very good point, and actually I think we should incorporate this into all of our meetings. Let me just give me a second. I have to check if the stew is not burning. I'm back. Yeah, so this is I think a very important point, and I think we should have this in mind in all of our works. So finding those points where we can check, for example, if you lost half of your data, which you don't always realize, I think. So if you have a raw data with 1 million lines and somehow your next file is only saying something about half of it, then you should know how to go back and how to check where the other half is. So, but of course, this is different from in every different pipelines and workflows. I think this is why it's very important to understand what is happening behind the pipeline. It's not just running it, it's so convenient. Usually just copy and paste the comments from the pipeline website and do what they say. But if you understand, and I think this is what we will also have in our minds when we are having these meetings, understand what is happening with the input during the pipeline, and then you will know what to look for in your temporary files or final results. I promise I will take this as a very important point, but also I'm asking you to always just to remind me that this is a very important point. Thanks very much. This is a very good thing. We will do your metagenomics quite early, and you will see how this is going. Any other ideas? Okay, if there is no more, that's okay. I think it's this is where we will stop today. I wasn't able to fill up all the 2 hours today, but I'm sure it won't be a problem from now on, because we will have so much to do. Please go on the server, try anything you want. You can install programs, you can copy your data and try working on it. Just try to use it. Be very comfortable to use it. I will try to write the next topic as soon as I can, the email, because I will also give you the links to the different editors you can use for writing scripts. But actually, if we will write R scripts much more, then we will use RStudio anyway. And I think, yes.
I have another quick question. So is there a way or a command where you can actually check quickly which are the softwares that are already installed in the server, or you have to just use it, call it, and see whether it appears or not?
That's the easiest way, just to write the program in. So there will be a few cases when we will download something and just unpack it. So as you may remember last week, for example, when we installed Metaspades, we don't really install Metaspades. We just downloaded it and we had it in a folder. So what we can also do, I think it will come together with the work we will do on the server. That, for example, we can have a software folder in our home directory. So if you just go back to your home directory and you will list what is in inside the software directory, which is visible for all of you because you are logging in with the same username, and you see the program there, and you don't have to download again. So this is again something what we can get used to, just not to download software everywhere. If you want to download a program, then put it in the software directory. If you want to download a data file, you should put it into your own folder in your Scratch, in the Scratch main folder. We already spoke about this today, but I think as soon as we are going through the meetings, this will come up every time, and it will be just a normal habit for all of us, all of you. So I don't think I have to write this down, but this will be something what you will see and practice.
Thanks.
There are a few cases when, so when we are installing something with Conda, so there are two ways you can do Conda installations. The Conda can install things in your main environment where you are at the moment, or the Conda can make a separate environment for that program. So there is a chance that, for example, I don't know what was the problem, but let's say that, for example, Bowtie needs a separate environment. So when you are logging in to the server and say Bowtie, there won't be any Bowtie or working Bowtie version up on the server. But if you are going into that environment, then there will be. So I think, again, maybe this should be a kind of like habit to have a txt file in your main home directory when we will collect what kind of environments we have, because this is something what I don't know if there is maybe there is a way to list environments. Let me check it.
Yeah, there is a command that I don't remember now, but I have checked all the environments that I have in my Linux, and I'm sure there is a command that will just list it.
Oh yeah, there is a conda info --ens. So this way you can also check if there is that kind of environment. But I don't know how. We will check this later, because I don't know how detailed this. For example, if someone is just giving PY27 as the environment name, that doesn't give you too much information, just only that it's a Python 2.7 environment, but you don't know what kind of program is in that environment. Like, I can imagine that you can also get a more detailed list or